Neurons are nerve cells that originate, process, transmit, and receive nerve impulses. Its basic structure consists of a cell body, multiple dendrite branches, an axon where signals flow through, and a synapse in which neurotransmitters are released. Ions like potassium and sodium cannot diffuse through the phospholipid bilayer. Protein channels embedded in the membrane are required to transport the ions in and out of the nerve cell. There are a greater number of potassium pumps than sodium pumps, making the membrane 100 times more permeable for potassium ions. More potassium ions are found on the inside of a nerve cell, and more sodium ions are found on the outside of the cell. The membrane of a cell is positive on the outside and negative on the inside because a greater concentration of positive ions are found on the outside. A typical nerve cell has a membrane potential of about 70 millivolts. When a signal is received by a dendrite, the signal opens some sodium channels and allows sodium ions to flow into the cell along its concentration gradient, which depolarizes the cell. If the cell membrane potential reaches minus 55 millivolts, the threshold, an action potential is triggered and opens most sodium channels, while the potassium channels remain closed. The action potential spreads to a neighboring region of the membrane and initiates another action potential there, and this process continues. The signal moves in one direction, from the cell body to the synaptic terminals, and after the action potential leaves a region of the axon, the membrane is repolarized when potassium ion channels open, allowing potassium to flow out. During the repolarization process, many potassium channels remain open while sodium channels close, resulting in a more polarized membrane than its resting potential, called hyperpolarization. To further balance the concentration of sodium and potassium ions, the sodium-potassium pump transports 3 sodium ions out for every 2 potassium ions that it transports in, again creating a net export of positive charge. To summarize that process simply, the opening of sodium channels result in an increase in membrane potential, and once that potential reaches the threshold, the curve's rapid increase represents the opening of many other sodium channels, resulting in further depolarization of the cell. When the potassium channels open, the membrane potential repolarizes and dips even further below the resting membrane potential in a process called hyperpolarization. Lastly, the sodium-potassium pump further balances the concentration of ions and returns the membrane potential to its resting potential. However, certain substances disrupt this process, such as tetanus toxin. The neurotoxin tetanospasmin, an agent that causes tetanus, targets the synaptic region during this process of signal transmission in the central nervous system, specifically a CNS inhibitory interneuron. It can originate from infection of Clostridium tetani, a type of bacteria common in soil. The toxin travels from the peripheral neurons to the central nervous system via transcytosis. Once the toxin reaches the synaptic region of the target neuron, it cleaves the V-snare protein, also known as SNAP25, needed for vesicle endocytosis from the presynaptic membrane. This effect targets neurotransmitters such as GABA and glycine. Because it targets the central nervous system, it can cause numbness and even spastic paralysis.